For the next fairly large series of lectures, we want to move back into molecular genetics. We have been off on transmission genetics and cytological genetics, and we talked early on about some of the molecular genetics. We talked about DNA structure, but now we want to move back into really thinking about some of the basic parts of molecular and apply. Okay? And we're going to think about the terms of getting from a genotype to a phenotype. And we're going to do that by thinking about expression and through the process starting at transcription. And the reason we're doing this, as you may remember with the definition of genetics that I used early on, it's a branch of biology that deals with heredity. So we've looked at heredity through these movement of genes, alleles, and the modes of inheritance and all of those rules that we've used, okay, for transmission genetics. And now we're going to talk about the expression of inherited traits. So we're talking about expression now because expression falls under this area of molecular genetics. Okay. So this is what's known as classically the central dogma. It's basically a model of how we get from genotype to phenotype, meaning that we start out as DNA and we eventually end up producing proteins. The idea being, again, that there are no noses or ears produced, that we don't have genes for those. We have genes that produce proteins that are assembled into these various organs or tissues types. The process involves taking DNA as a blueprint code, reading the information off of that, converting that information into RNA, and then RNA is taken in various forms, and it's usually, and if we're talking about a eukaryotic system, moved to the cytoplasm where it is translated. The information on the message is translated. Ultimately, it's going to be making protein. Now, this is not a straight shot. We're going to see that we go off in various directions, okay? Now, I'm not going to spend a huge amount of effort on each little step talking about all the details because at this level, our job, we've been tasked with the job of sort of thinking through the whole process and making sure you have a good picture of just what happens in this movement from genotype to phenotype. Other courses you'll study a lot about transcription, translation. In fact, advanced courses, you can take an entire semester just covering these two ideas and talking about transcription factors and the development of all the elements needed for translation, etc. And certainly if you take biochemistry, cell biology, advanced molecular biology, you'll spend a great deal of time on all of these. So don't worry about the fact that we're sort of cartooning it and not filling in lots of details. In fact, we're not even filling in all the details that you'll probably find in a basic textbook. But I want you to understand the process. That's what's important for us. So what are some of the basic rules? Well, first, transcription is obviously a selective process, unlike replication, where we just start out at a particular replicon point and we just replicate everything from there around there. Obviously, we don't want to do that. We don't want to replicate an entire or read an entire chromosome and transcribe it. So only specific parts of DNA are transcribed, meaning genes, right? Transcriptional units. Okay. The process involves taking single-stranded DNA. So we must take our double-stranded DNA and open it up. And then from that, read the information and produce RNA. Okay? So we're going to produce RNA from that. Generally, only one strand is transcribed and certainly at any one time. Now, you may remember that I showed you some examples of where there were genes where one strand of the DNA represented one gene, and then sometimes they overlapped with another gene, another part that was on the other strand. But when a transcriptional process occurs, it usually only involves one of those strands or the other. So only one gene at a time is used. Okay. The RNA messages are obviously, because we're going to read, we're going to start at the 3' and produce a 5' message. So what's going to happen, it's going to be anti-parallel and complementary to the DNA. The only difference will be that we'll replace the thymines with uracils, and because we're dealing with RNA, as well as, of course, you have a slightly different sugar, 5-carbon sugar that you'll be using because it's RNA, not DNA as well. Transcription always works 5', 3', but what that means is the first part of the message that's produced is the 5' end. So go back and think about the structure of DNA. It's been a while, but think about the structure of DNA and what 5' means. It's a 5' carbon versus the 3' carbon and how we add on to that structure. Okay. The process involves what's known as RNA polymerase. We're going to just use a generic term for that. In reality, it's a large enzyme complex of multiple types of enzymes, but the base idea is that they're going to bring in these triphosphated nucleotides, A, UGC, and they're going to link those together with forming phosphodiester bonds. Okay, so it's a big complex to bring those in to catalyze that reaction, etc. We're just going to call it RNA pol. There's different types of RNA polymerase, as you'll learn, and more about this complex. And then to make all this work, we obviously need these places to indicate this is a gene, right? This is where we need to go, not only to start the process of transcription, but also to end the process of transcription. And these little signals are called motifs, and they're critical for binding of the RNA pol to the DNA, for signaling where you want to start, where you want to stop, etc. Now, we've looked at some of these motifs. Okay? We've talked about TATA boxes. We've talked about some of these other cat boxes and other promoters as well. But we also need to be aware that we're going to talk about things like a polyase signal. Toward the end of your transcriptional unit will be a site where there's a signal saying this is a polyA tail site, common. And then there's usually other signals in there. And then the end, of course, is GC rich in some form, or GT rich, depending on which ones we're working with, but usually GC. And so you have very specific signals to say this is the start of a transcriptional unit, and then somewhere down here, oh, you're getting toward the end of this gene, and we need to start activating the system to stop the process of transcription. All right, so we're primarily going to concentrate on TATA boxes as our initiator up here, but you need to realize there are all sorts of initiator elements and motifs that are involved, and those are fair, again, another series of complexity there beyond the scope of what we really need to worry about. Okay. All right, so we're going to basically break down transcription into 4 steps. First, there's going to be a recognition step, which is the pre-initiation. In other words, getting everything together, right? Before you start to build anything or to do even write, a paper or something, you have to pull all the information down and get it in front of you so you can work. Or if you're going to build something, you got to get all your material together. So that's sort of the first part. We'll call that recognition. Start pulling things together, finding the right site, getting everything started. Then we'll initiate the process, okay, which basically involves organizing this RNA complex and getting RNA polymerase active. Then we simply move along and elongate through the process, making more and more message. And eventually we have to terminate, right? We have to stop the process. So we have to cleave off the transcript, get it loose from the DNA, get it separate, get that DNA back together. And then we've got that message that we can start to manipulate. Okay, I'm going to go through each of these. This first little part lecture is really about recognition and the pre-recognition, and then we'll organize the second lecture into initiation, elongation, and termination. We'll make those parts all fit together in one lecture. So with recognition, well, think about what DNA looks like, right? We've defined chromosomes as having chromatin, nucleosome. So we're out here in G1, right? We're out in the normal phase. We're making most of our proteins. Our DNA is at least beads on a string, at least these 11 nanometer fibers. It may be 30 nanometers. It could be all sorts of things, but certainly the DNA is just not naked strip of double-stranded DNA. It's coiled around all these nucleosomes and things. So part of the pre-recognition process, or the recognition process itself, is somehow you've got to get into an active phase. Remember that once again, euchromatin and heterochromatin are sort of they're a continuum, right? And with heterochromatin being where it's fairly tightly packed up, euchromatin being where it's more loose and available. And so they go back and forth between those. So we have to have ways to do that, right? So we have this, if it's in a heterochromatin state, then it's silenced. We can't really transcribe it. We've got to get it opened up over here and get it into a state where we can actually function, where the machinery can come in and take place. So we've got to form at least balls on a string level to make it work, okay? So what we're basically saying is that DNA must be opened in order to make it work. How this actually occurs and all this process and is becoming to light more and more and more every day is the understanding of what's known as gene expression. And we will talk and give several lectures about gene expression and gene regulation, how we actually turn on and turn off those genes. We'll talk about that a little bit later, but for now, realize that it's something we have to do, okay? Now, it has to happen in order that we can get our RNA polymerase down somewhere near the TATA box. That's what we're trying to do, okay? We're trying to get the RNA pole, this complex, recognized and finding this particular place that we want and starting to open up the DNA and begin the process, right? And here's a TATA box shown kind of in our little beads on a string right here, right? And if we take that TATA box and think about it, this is the gene going off down through here somewhere, right? And so what we want to think about is, remember these nucleosomes have these tails, right? We said that when you think about nucleosomes, you think about the proteins that make them up. They have these little tails that stick off to the side of them, and those tails are what then control, at least their chemical state controls whether the DNA is tightly bound into the nucleosome and the nucleosome is a unit, or whether it's able to release. Okay? And you may remember that that's done through several mechanisms, but the one that we illustrated was using what's known as HDAC. Okay? This is a histone deacetylator, so we knock off the acetylation, and we bring in a number of complexes of other things that basically bind the DNA up to make this form. Okay, if you want to open it up, then what you use is HAT, which is acetylators, and you come in and you acetylate those tails. Once you acetylate those tails, it starts to loosen that DNA from the nucleosomes themselves, change some of that charges from there, and also allows these other regions, these promoter molecules, to come in, and ultimately RNA polymerase to come in, and transcription process can proceed. Right? So let's think about recognition. Okay, so recognition is the formation of this complex, right? And this complex is recognized here as is PIC, right? So we have this large complex that and PIC stands for pre-initiation complex, and that initiation complex involves a number of proteins. All these things down here that have names on them, other than TATA, are proteins, right? So TATA is here, okay, and TATA, we have a couple of proteins that come along and bind. The first one is known as the TATA binding protein, TBP, and it will come along and find TATA that we're looking for, okay? And it will bring in some other binding proteins, which then themselves will start to pull in others. And once all those are in, they'll start to recruit all these general transcription factors. And when you see the word factors, we'll use that a lot. That just means it's a proteins. They have various names or forms, and they may be enzymatic, they may be structural, whatever. These are generally enzymatic type proteins, but factors just mean they're proteins. Okay. Now, actually, sometimes factors can also be RNA and other things. So it's a generalized term, but what it means here is we're going to build this complex. Okay, we're going to build this initiation complex. So we're sitting down on TATA, we're pulling all these elements together. Once they're in place, then they're able to recruit RNA polymerase, this larger complex, to pull in and start the process. Okay, so all this has to happen. As part of that initiation process, you get things like helicases, which come in, as you can see here, and start to open up the DNA, because we need to make it single-stranded, right? We need to be able to read it. So they actually start to pull the DNA apart and allow the process where we can start to read it. Also, you've got to think about all these nucleosomes have to be knocked off. The DNA is spherical, so it has to be opened up and pulled away. So the preparation for all of that has to occur in this pre-initiation process. Okay. All right. At this point, we've got recognition going on. We've recognized the site of TATA. We've recognized the gene that we want, which is another series of issues. And now what we're going to do is start the process of bringing in RNA polymerase and initiating the actual production of the message. And that will start out in the next lecture.